Well, good evening, everyone. I see 7 o'clock on my computer here, so we'll go ahead and get started. We have a few folks rolling in here in the next couple minutes. But welcome to part 3 of our Sheep Genomics webinar series. It's great to have everyone on again. We have one additional webinar coming up next week to conclude the series, and so I encourage all of you to get registered and sign up and attend next week, next Tuesday evening for that. Tonight we have Dr. Scott Greiner with us. Dr. Greiner is a professor and extension animal scientist at Virginia Tech. His name is probably familiar to many of you. He serves in both a beef cattle as well as sheep extension role, and for a lot of his career is focused on applied genetics and selection programs. He serves as the educational advisor to the Virginia Beef Cattle Improvement Association, as well as overseeing the Virginia Bull Tests and their Master Cattlemen's program. He works with the Virginia Ram Test program, and we really appreciate having Dr. Greiner on with us this evening. And so I'll turn the floor over to him, and he's going to discuss this evening some of the applied ways that we can utilize genomic tools, genomic-enhanced estimated breeding values in our selection programs.
Well, thank you, Andrew, and thank you to you and the committee for the invitation to take part in this webinar. I have thoroughly enjoyed the first two sessions, and hopefully what we visit about tonight will complement those and help us further tell the story and understand how this technology we call genomics can benefit us in the sheep business. It certainly is an exciting time to think about that. And maybe as a little bit of follow-up to Andrew's introduction, you might have heard that. And for those of you that don't know me, probably think, man, he does a lot of things. And I do. And so I'm going to approach this talk wearing lots of hats as well, both from the academic standpoint, as well as my role as in extension, and then working directly with our Katahdin flock at our Southwest AREC and our Dorset and Suffolk flocks here on campus, all of which are enrolled in NSIP. And so I kind of can wear various hats and going to approach this a little bit from that context. So Andrew, I don't know if you want to handle questions as we go and watch the chat box, or we do that at the end, or maybe a combination thereof. We can do whatever, but thank you all for joining us tonight, and again, appreciate the opportunity to be here. I thought I would start maybe, I'm sure lots of you were on our first couple of webinars, but just to get us all on the same page, and I've got a couple slides kind of from an introductory standpoint. Rusty and Dr. Lewis did an excellent job talking about various facets of this topic relative to genomics and genomically enhanced estimated breeding values for the sheep industry. So I don't want to repeat a lot of what they said, but at the same time, if you happen to not catch those, some of what we're going to talk about, I think is fundamental in terms of having the big picture view. And I very much take a big picture view in terms of trying to explain this, because on a daily basis, I'm not in the weeds, so to speak, at the academic side in terms of genomics, but more on the applied side and how we can use it. So I like to think of it in very simple terms as we're using that animal's DNA and variations in that DNA to help us further explain that animal's breeding value. Put another way, we're quantifying the DNA and turning that in part into an EBV in the sheep industry. And that's a pretty complex process that Dr. Lewis did an excellent job explaining and making it seem fairly simple, but it is ultimately really complex, and I think we all understand that. Maybe in a little more detail, what that really means is if we consider a reference population of animals, and most of the time we think about a reference population being a breed, and knowing that with the current Katahdin project underway, that is currently happening in the Katahdin breed, where they have a reference population of approximately 5,000 animals. Those animals have had DNA collected. That DNA has been analyzed and genotyped. And along with that, just as importantly, those animals also have phenotypes. They have performance records for the traits that we're interested in measuring, everything from birth weight to the weaning and post-weaning weights. In some cases, perhaps some carcass records, some scan data, maternal data in terms of number of lambs born and weaned, the fecal egg count data, and we can think of others as we think about other breeds and we think about other traits that are of interest to us. We then take that reference population and we use their DNA to analyze those SNPs and look for SNPs, which are regions in the genome that are of interest to us that might be associated with differences in performance or variation in performance in that population. So we're really using the reference population as our template, if you will, to understand what differences in DNA or differences in DNA variation in these SNPs specifically. How informative are they, and what do they tell us about the performance of those animals? Okay, so then we can take an outside animal, and I'm using Katahdin here as my example. So our Katahdin reference, we've got a Katahdin ram who's outside of that reference population, but he's in the same breed. He's genetically related to a portion of that reference population. We can genotype him, collect his DNA, perform that same analysis, and use the reference data that we've trained and learned from to inform us about what his DNA and what his SNPs mean relative to his genetic merit. Okay, so that's kind of a simplistic way that I like to explain and think about it. We then take that result from that Katahdin ram, we incorporate that into his EBV calculation, okay, along with this pedigree data, along with his own performance. If he gets to that point and has progeny as a ram, as a sire, that would be included as well. And a component of this also, as Dr. Lewis talked about last week, is establishing his genomic relationship pedigree-wise to the reference population and his relationship to animals in his pedigree and how that relates. And we do all this simultaneously to calculate and come up with a genomically enhanced EBV. I've heard people use the analogy, and I think it's a really good one. If we think about the gas, if we think about EBVs being a vehicle or a tool, a truck, if you will, we might think about the gas and the oil being the pedigree and the performance records. They are key to making that EBV viable and making it work. And we might think about the genomic component being a fuel additive, okay? If we use fuel additives, hopefully it makes our vehicle work better. Genomics can complement what we're already doing in terms of NSIP, in terms of EBV calculation. And so it's a piece to the puzzle for sure. And tonight we're going to kind of concentrate on how it can help us some various ways and some tools from that standpoint. So Andrew gave me the title, how does it help us with selection? I don't know that it necessarily changes our selection per se. What it does is it enhances the tools that we have at our disposal. And we're going to touch on a few of those things as we move through this evening's presentation. So I'm going to start by kind of as an overview of what I'm going to describe. I go back and think about how might genomics enhance our ability to make selection decisions, or how might it enhance the tools that we're using? And if we think about how genetic progress or genetic improvement happens, there's 4 essential components to that. And many of you have seen this or remember it from back in your days in college. We probably don't think about this when we're selecting rams or selecting ewes, but it's kind of the principles under which we operate. So the accuracy of selection really has to do with the relationship between the estimated breeding value that we have on an animal. In other words, the EBV that we get back from NSIP, what's the relationship of that to what the animal's true breeding value is, or an accuracy of 1, if you will. We never know with 100% certainly, or with an accuracy value of 1, what that true breeding value is, okay? It ranges from 0 to 1, okay, on lots of different things, but genomics certainly has an opportunity to help us there, and we'll visit about that. The intensity of selection has to do with the superiority of the selected animals compared to the flock average, okay? In other words, how much reach do we have? How much better is that ram's EBV compared to our current flock average? How far can he take us in terms of improving a trait that we're interested in or is economically relevant to us, okay? Genetic variation has to do with how much change can we make, and that's contingent upon variation in breeding values in the population. So if we want to take a particular trait a certain direction, okay, how far we can go is dependent on how much genetic variation is at our disposal that we can select from, okay? So we'll talk about the influence of genomics on all these factors. And then the generation interval, which is simply what's the average age of the parents when the offspring are born, okay? In sheep, we generally think about generation interval being somewhere between 3 to 5 years. That can vary depending on age of your rams, which are generally younger than are the ewes, how fast we turn the flock over in terms of ewe replacement, use of some technologies like embryo transfer, et cetera, can enhance our generation interval. But that's the other component that's involved in our rate of change as well. So we're going to go through each one of these and talk about some practical applications as specific to genomics, and how genomics may or may not influence these factors that influence how we make genetic progress and how we approach things. So we're going to start with accuracy, because in my mind, that genomics probably has the biggest influence on accuracy, and it does so in several ways. The first of which is parentage verification. And as Dr. Lewis talked about, part of the genomic process is verification of the sire and dam. And of course, when we think about EBV calculation, the pedigree is the foundation, okay? And I wanted to work through a little bit of a very simple example to kind of illustrate this. It's something that I don't know that we think about a lot, but it's pretty foundational in terms of how EBVs are calculated. So through NSIP, of course, for most of us and most flocks that are enrolled and have access to EBVs, both the sire and the dam have an EBV, okay? And we know that we can simply average those two, and that's the pedigree estimate of the resulting progeny or the resulting lamb crop. In this case, if the sire is 4.5 for post-weaning and the dam is 1.5, by pedigree, that lamb that's born is 3.0, okay? Of course, we use this on a regular basis when we make mating decisions. As we make corrective matings, we look at the EBV profile and the genetic merit of the dam. We find rams or sort ewes, if you will, into ram breeding groups, largely based on this particular principle. And so it's pretty fundamental that, number one, the sire is correct, and number two, the dam is correct, because in terms of calculating an EPD, that's where we start, okay? And from there, of course, we build on that with the submission of individual performance data on those lambs. That EBV can then fluctuate based on that animal's individual performance. In this case, I have that being favorable and his EBV actually increasing, at least for more growth. As you understand, that could go the other direction. And of course, we've got things working in the background here. We've got this animal's relatives, his half sibs, animals in his pedigree. So it's a continually evolving process relative to that pedigree as we gain more information from our flock or perhaps other flocks that have relatives that contribute ultimately to this EBV. And then ultimately, if that animal goes on to be a parent, a ram, for example, and sire's progeny, that's going to influence his EBV, of course, in a significant way. we tie all this together and we include the genomic component. And one of the principal things as I view it, in terms of helping us from a selection process and the accuracy of selection, the first of those is establishing that this pedigree is correct. Okay, genomics enables us to sire verify. So let's take, for example, in this case, that through genomics and through DNA, we find out that in fact the sire that we have submitted as the pedigree of this lamb, in fact, is not his sire, and instead the sire that he's verified to has a post-weaning EBV of only 2.5. Okay, that obviously is going to influence what happens, because his post-weaning EBV suddenly by pedigree becomes 2 instead of 3. Depending on what breed this animal is, that could be a significant difference in terms of how we evaluate that ram in terms of his genetic potential, either favorably or unfavorably. Okay, we could have used the same example that perhaps the sire that he's actually, the ram he's actually sired by has a superior EPD to the one we thought he was sired by. But in either case, the principle is that that we're confirming that in fact our pedigree is right, which forms the foundation of our EPD calculation. And then just as we've shown before, we add his individual performance to that and his progeny performance. So I think a very important tool. I've only shown the sire example here. Of course, we can also verify the dam, that the dam is correct. Both of those are possible and fundamental in terms of how we use genomics and how that will be incorporated. So why does this happen? And I think as we all reflect and think about our own flocks, there's lots of ways this can happen, and how the sire or the dam can be incorrect, and simple misidentification of rams. Those rascals are ornery. We've all had rams jump the fence. We've all had rams get together, or breeding groups get together, and somebody forgot to shut the gate. Those things happen. And it becomes, rather than a single sire mating group, it becomes a multi-sire breeding group. And that can happen for a short period of time or an extended period of time. And so those things happen. We've got mismothering at birth, which makes the sheep species relative to this particular topic a little unique compared to cattle. Because of multiple births, mismothering, we know that happens. And lastly, there's human error involved. All of us, as we make those records in the barn and we put those in our notebook, or however we choose to do that, we then take those to our computer, we enter those into NSIP. So we've got 2 or 3 or 4 different opportunities there to have a recording error or a transfer in data error, however you want to term that. It certainly happens. So to shed a little light on that, Dr. Lewis shared with us in the process of collaborating on the Katahdin project, the sire and dam verification results on our Virginia Tech Katahdin flock at the Southwest AREC that Lee Wright looks after. And so I thought I'd just share that data because I think it kind of sheds some light on this particular topic. So we genotyped 223 total animals through the project. 187 of those animals, we had the potential to sire verify them. Okay. The other ones we were unable to. My assumption is we didn't have DNA on the sire. Of those 187 that we had sire verification information on, we confirmed 177 of them. So right at 95% of them were confirmed to the sire as recorded. 10 of those, or 5%, were not confirmed to the sire. Okay. And so we had an error rate of 5%, if you will, in terms of sire verification in the pedigrees of those 187 lambs. Now, we were only able to pedigree verify because of the structure of this particular project. There was only 23 of them that we could possibly verify to the dam. 3 of those 20 we weren't able to verify, so 13%. Okay. which seems like a relatively high percentage, but my speculation would be is that's not totally out of bounds in terms of what we might expect when you consider mismothering opportunities and all the things that can happen in the lambing barn, etc. So I think that gives you a little sense. I think we know from the beef industry that parentage verification errors certainly happen, whether that's through recording the wrong straw of semen or pulling the wrong semen out of the tank. Lots of different opportunities that can happen, or in fact, it's not an AI calf, and in fact, it's bred by the cleanup bull. Okay, we have those same examples that we can talk about in sheep. I'm happy to report, though, that 100% of the time we got the gender of the animal correct, which honestly I expected perhaps we'd have one or two errors there simply based on data recording, but they did gender verify all 223 of them. One other thing I wanted to follow up on, and Dr. Lewis shared some information on this last week, was a little bit one of the tools and one of the opportunities that genomics allows us to do is to establish genetic relationships between a particular animal and those in its pedigree as established through the reference population. So typically when we think about a pedigree, and in this example, I'm only considering the sire. So we've got this sire here, and the green bar underneath the sire represents the half of the genes that he inherited from his sire or the paternal grandsire. The blue bar represents the half of the genes that he inherited himself from his dam or the lamb's paternal grandsire. Okay, so what we expect based on pedigree is that this lamb will inherit half its genes from the sire, which in fact it will, and half of its genes from the dam. But what we assume by pedigree is that the half of the genes coming from the sire, half of those, or a quarter of the lamb's DNA, if you will come from the paternal grandsire, and a quarter of it from the paternal grandam. Okay, that's what we expect on average. But we know through genetics and Mendelian sampling that doesn't always happen. We know we get variation through various processes, recombination, that, as shown by lamb number 2 here, he actually inherited a higher proportion of his genes from his paternal grandsire than he did his paternal grandam. Okay, we can flip that around on lamb number 3, and it might work the exact opposite or the inverse of that. And then we've got everything in between. Okay, on average, that's a quarter for each one of those relatives, but in individual animals, there can be a significant range. And so one of the powers of genomics is that it allows us to identify this and actually track or trace this through that animal's pedigree to determine the proportion of his genetics that is inherited. And we can use that to our advantage then in terms of calculation about his with his EBV. Let me show you a cattle example that I think is interesting and kind of illustrates this point. And this is from the Angus Association and presented at the Beef Improvement Federation meetings a couple of years ago. So I've borrowed that and used it on a few occasions. So there's 2 full sib brothers, actually embryo transfer brothers. And so they're both sired. Of course, by nature, they have the same sire and dam. So their pedigree relationship is they are full sibs. Okay. In fact, one of those calves has a birth weight EPD of 1.6. One of those calves has a birth weight EPD of 2.5. Okay. So what was done in this little example is they tracked the pedigree and the genomic relationships between these 2 full sibs back through the pedigree to the male ancestors in the pedigree of those animals, okay? The pedigree of the two animals is the same, of course. Okay, this just happens to show the top side of his pedigree. So through his pedigree matrix, we can determine, on average, what is the relationship between this animal or this calf and each one of those sires in his pedigree. For example, that sire at the top, Shearbrook-Shoshone, that relationship is 1/64. So 1/64 of this animal's genetics relate back to Shearbrook-Shoshone. And we can do the same thing for all those sires in this animal's pedigree, okay? So we can do that on paper based on the pedigree information, all these sires that you see listed in this table, okay? Then we can use the genomic relationship as determined from the genomic pedigree, okay, to determine what that relationship actually is, okay? Each one of those sires, many of you familiar with the Angus breed will recognize most of those sires' names. They are highly proven, popular, well-used bulls in the Angus breed with established EPDs, and so we have the EPDs on those bulls. So what they found that was interesting is the calf that actually had the higher birth weight, EPD himself, was 15% more related to the higher birth weight sires in his pedigree, okay? In other words, he inherited more genes from the animals in his pedigree that were higher birth weight than we would expect him to based on his pedigree, okay? In a similar fashion, that same animal with the higher birth weight EPDs also inherited 13% less of his genes from the low birth weight ancestors in his pedigree, okay? So based on that, that explains why his EPD is higher based on his ancestry as documented through genomics. So that's one of the powers that we have. And Dr. Lewis mentioned that last week, and we use that in combination with the SNP information to help us describe this animal's pedigree, which really helps us in terms of that EBV calculation. And this is kind of a real world example, it being beef, but the same principle applies as we talk about it in the sheep world. Okay, relative to accuracy, let's think about some traits that are important to us, and talk a little bit about how genomics might influence some of these particular traits. One of those traits that we think about that genomics having a favorable influence on are traits that are expressed in one gender, okay? And I think the perfect example in the sheep world is prolificacy or litter size or number born, if you will. That's a very important trait to us economically across all breeds. We know that that's only expressed in the female, right? Only ewes express that when they lamb. We can't measure that in a male, okay? So genomics can help us using that particular structure that we visited about in terms of those animals that don't actually have a phenotype for that particular trait of interest. Genomics helps us through use of those SNPs and that pedigree matrix analysis, helps us to do a better job describing the genetic merit of that animal when in fact we don't have a phenotype on that particular animal for that trait. In a similar fashion, we have traits that are measured late in life, and Rusty mentioned some of these in our first presentation. Longevity, in other words, how long does that ewe stay in the flock? How long is she productive? In other words, does she wean lambs and rebreed and stay in the flock, or does she remain open? How long does she stay in the flock? That takes some time for us to accumulate phenotypes and accumulate data on, okay? But utilizing a proper reference population structure with adequate numbers of phenotypes, we can use genomic data to help us enhance our accuracy and enhance our ability to describe these traits that are even measured later in life even on young animals, when of course they may be several years or some time away from actually having a phenotype for that particular trait of interest. Expensive or difficult traits to measure, we also know it can enhance us. And we think about the classic one of those is feed efficiency. If we draw the analogies to the cattle world or the cattle industry, feed efficiency is one of those traits that they have begun to focus on, and genomics has been an important component of that. And of course, that might also, a trait like feed efficiency might also fall into the category that it's only measured on a select handful of animals. Typically, we think about that being measured on terminal animals, maybe in a feedlot setting. Perhaps we can measure that in breeding age bulls. We typically don't measure it in females. And so it might have some limitations there as well. Other things that we might think about down the road that are expensive and difficult to measure, things like tenderness, eating quality traits, and those type of traits as well. So I think part of the thing that we need to discuss as an industry as we think about these expensive or difficult traits to measure is how are we going to go about if we decide that these are economically relevant and we need the selection tools to enhance those within the sheep population. How are we going to structure a genetic evaluation that we can go about measuring these traits in such a fashion that when we collect that data, it can have the most use and have the most utility across that breed or population. An example of that we might think about is perhaps we have collaborator traits or collaborator flocks, excuse me, that we've designated within a particular breed or population that we're going to measure feed efficiency and perhaps some of these end product traits on. It will be very important that those collaborative flocks represent the population in general. And if we think about that as a breed, make sure that the genetics within those flocks represent the breed that we're going to imply those measurements and the genomic tools to make sure they're representative in terms of their genetics and their pedigrees. So I think that's one of the things we have to think about from an industry perspective is how are we going to tackle this relative to some of these traits that are expensive or difficult to measure. Number one, deciding which ones they are. And then number two, how do we structure it in a practical sense that we can collect enough data, both genotypes and phenotypes, that it will do us some good and have utility moving forward to advance the entire industry as opposed to a small segment of it. And then lastly, the lowly heritable traits. And we think about lowly heritable traits like reproduction or fitness traits, where a single phenotype or an individual measurement on an individual animal, while informative, it is less informative than a phenotype on a more highly heritable trait. So we can think about the genomics being able to couple with the phenotype of those lowly heritable traits and help us provide a better estimate and strengthening our ability with that lowly heritable trait and the measurement of a single phenotype. We can couple that with genomics to strengthen our ability to make that genetic prediction as well. Okay. One thing I wanted to mention, and we've touched on this already as we think about the sex-limited traits, but having genotypes on females is very important. And I think that's one of the lessons as I've worked with colleagues in the beef industry that they have learned over the last several years, is that having genotypes on females is very important, okay? If we think back to what the cattle industry did, largely in the early days, genotypes and collection of DNA was largely focused on the male side, on sires, because of, and we think about that intuitively that might make sense because of the profound impact that the sire has on a particular herd or flock, much more significant than an individual female. However, we have so many traits that that males don't have phenotypes in, and we've talked about several of those already. The reproductive traits, the prolificacy, number of lambs born, etc., that we only measure in the females. Okay, so as we begin to build reference populations across breeds, and we think about that, it's very important to have both phenotypes and genotypes on females as well. Okay, and that would include mature ewes that have multiple lambings, so that we have those phenotypes across several different parities. Okay, ideally we would have also genotypes and phenotypic records on ewe lambs, so we can use those early records, growth if you will, weaning and post-weaning, and perhaps ultrasound records early in that ewe lamb's life. But if she turns into a dam and becomes a productive ewe, she has a lifetime record in terms of phenotypes or production records that we can associate with her genotype or her DNA that helps us from a referenced population standpoint. So think about the females as well as the males. It's easy to get to not think about females because of their relatively small contribution individually to the genetics of our flock. But in terms of genetic prediction and utilizing this technology, they are extremely important for those reasons. So we talked about selection intensity being important, and that's the superiority of animals selected to be parents. And I think one way to think about this or illustrate that, we think about the percentile tables that NSIP publishes and makes available to all of our breeds. These really define for us selection intensity. So if we take an individual animal and his EBVs and we look him up on this particular table, we can establish his superiority or inferiority, if you will, relative to the population in that breed. Okay, 50th percentile, of course, is breed average. And we think about a Katahdin ram with a post-weaning weight EBV of 10. He's in the upper 1% of the Katahdin breed for that particular trait. Okay, so he has his selection intensity would be potential would be strong based on that. But of course, that has to do with selection intensity per se is not really impacted by genomics. What is impacted is our ability to accurately define that animal's breeding value, and therefore more accurately define animals that are superior, which enhances our selection intensity. Okay, so really the impact on selection intensity goes back to the accuracy component. Inherently, we're not going to change the breeding value per se by genomics, but what we are going to change is our ability to accurately assess the breeding value and therefore more accurately define selection intensity, which enhances our genetic improvement. Generation interval, okay? In short, the way genomics can help us from a generation interval standpoint is that we have better information, more accurate information, more accurate breeding values on animals earlier in their life. And this is kind of sums up lots of the previous slides that we've talked about, that being the enhancement of accuracy and the ability to have more accurate estimates of breeding values on traits that occur later in life. Okay, so I think there's a lot of value in this. And one way that I thought I'd present to put this in context a little bit, and I'm going to use the beef example again. Unfortunately, I don't think we have data at hand in the sheep industry yet that we can draw a similar, that we can create a similar table. Hopefully down the road we'll be able to, but the principle here holds true. Okay, so this is from the Angus Association, who's been in the genomics, genomically enhanced EPD calculation process for several years now. but they publish a table that relates progeny equivalence. Okay, so a better way to say this or think about it from an applied standpoint. So if we have a genomically enhanced EPD on an animal, let's say it's a bull, okay? By nature of using genomics and what genomics brings to the table in terms of that pedigree matrix, as well as the SNP technology, how many progeny would it take under normal circumstances using traditional EPD calculation methodology? What's the progeny equivalent to the genomic information? So you see several traits listed here, and the one I want you to focus on is milk, okay? That's on the right-hand side. So what this says is that if we have genomic information on a young sire, and we combine that genomic information into his EPD calculation, along with his pedigree, along with his performance records, what that tells us is that that genomic information and his genomically enhanced EPD is equivalent to having 36 daughters on record to calculate his milk EPD, okay? So I want you to think about that just a little bit. If you think about that in the beef context, in order to get 36 daughters with progeny that have weaning weights, because that's how milk EPD is calculated, just like in sheep, on a young sire, that's approximately a 3.5-year process, okay? We can have this genomically enhanced EPD before that bull even reaches puberty, long before he reaches puberty, and can even produce semen, okay? But in a traditional sense, it takes 3.5 years from the time that bull can produce semen or he can breed a cow until his daughters are in production and wean a calf. Okay, and we think about the economic benefit of that and what impact that has on our ability to make accurate selection decisions and generation interval. It's significant. Now, I caution that the generation interval is shorter in sheep. They have multiple progeny, and so the numbers are going to look different when we have the opportunity to reflect on this as it relates to sheep. But I think the principle holds true to kind of illustrate the point in terms of the potential of genomics and how it can help us in terms of more accurately describing breeding value and differences in genetic merit. One other way that genomics can certainly help us from a practical standpoint, and we're doing this already, is identification and quantification of various causative genes. Okay, and these causative genes might be for genetic disorders that are undesirable. They might be for genetic resistance to particular diseases like scrapie and like OPP. They might be as simple as difference between an animal being horned and an animal being polled. Okay, but genomics certainly can be a component of that. And like I said, we're currently doing this because most of these are controlled by a pair of genes or a relatively small number of genes, so they're causative and have a large effect on what we're measuring or what we're seeing in terms of phenotype. Okay, but I guess I wanted to throw out just for discussion and thought relative to this particular group. You know, we think about this and we can send off DNA currently to a variety of labs, and we can get measures for spider syndrome. We can get measures for scrapie susceptibility, everything that's listed on here. Okay, the difference being in the sheep industry, we don't have a repository that I as a breeder or you as a breeder can go to that shares the results on a particular animal as it relates to these particular genes or this particular DNA. Okay, I guess the exception I might say is that we do record on the pedigree or the registration certificate if they're horned or polled in some breeds. But all these other traits, we can do that voluntarily, but it's not in any way connected to the pedigree of the animal, that comes to us as a breeder or shared amongst breeders. And that's quite different than we see the structure of the beef industry, where we have the integration of that animal's pedigree documentation done by the breed association, much like we do it in the sheep industry. But that's directly tied to and associated with that animal's genetic evaluation and any particular tests that relate to genetics or genomics that might have been conducted on that animal. So for example, we can pull up the Angus Association website, plug in a registration number on an animal, and we know exactly how they test for any particular genetic disorder of interest, okay? We know exactly if they've had genomics done and if genomics is included in their EPD calculation, okay? We can do the same for Simmental. We can do the same for just about every breed. So that's quite different than the current structure of our sheep industry. And so as we think about advancing our genetic evaluation, I think these are things that we need to think about and things we need to talk about as an industry moving forward collectively. So what are some added benefits? Some things I thought about that from a practical standpoint, we all struggle with as it relates to performance recording, as it relates to calculation of BVVs. And here's a list of animals that we're challenged with. Orphan lambs, grafted or fostered lambs. And I know we've got some new protocols and processes involved with NSIP for foster lambs that came out recently. But these animals are challenged relative to their individual performance measure or their phenotype, okay? Genomics, by nature of having that tool, that phenotype, perhaps in certain cases, and I don't want to de-emphasize the need for phenotypes, but there are certain animals under which the phenotype is not as informative. Orphan lambs and grafted lambs, foster lambs are an example of that. We all have lambs that have contemporary group challenges. Maybe they're born late, they're born outside the window of the contemporary group. They're a single contemporary, and so their individual phenotype is less informative. If we think about the application of embryo transfer and recipient dams and how you account for those, again, these are all cases that I think genomics can help us in terms of better describing the genetic merit of these animals using that process. Animals with missing data or phenotypes, we simply missed a record. We didn't write it down. He got through the scale, whatever the case might be. And potentially, and I know this has been one of the challenges that I've experienced personally in our flocks here, is introduction of non-NSIP enrolled sheep into the system. Perhaps genomics can help us with that. If that animal doesn't have a direct pedigree tie, at least in terms of his registration papers, perhaps that animal does have a genetic tie to the reference population, albeit it might be back in the pedigree, but perhaps we can use genomics to help us better associate the genetic relationship of that non-NSIP sheep with the reference population. And perhaps we can use genomics and the SNP technology to help us better predict what his breeding value is, even though his pedigree is of limited value in terms of a direct sire and dam that's enrolled in the program. So another potential way that may help us, okay? Another cattle example real quick. These are two bulls that I've selected and bulls that were actually fed and managed together on our bull test program. They are not brothers, they are not related. I selected them for a couple of reasons. One is in terms of their calving ease and growth EPDs, they are very similar. We would categorize those bulls in terms of their genetic merit as being very similar. Where they are dramatically different, they are both embryo transfer calves. The bull on the left has an accuracy value of 0.05 for those traits of interest. The bull on the right has an accuracy value ranging from 0.33 to 0.48. That bull had genomics conducted. His EPDs are genomically enhanced. That's the bull on the right. The bull on the left is not. okay? So the take-home message is that the EPDs on these two bulls say that they rank in the top quarter of the breed for calving ease. We'd evaluate them both as calving ease sires. However, their accuracies are dramatically different. And how we interpret that is that the bull on the left, even though his calving ease is +10, which is very desirable, his possible change associated with that accuracy value means that his EPD could really range somewhere between +1 and +19, okay? That's a huge range, okay? A lot of potential error involved there. The bull on the right, that possible change is much smaller. That window is narrower. And so he's a better bet and less risky in terms of using him as a calving ease sire based on possible change of his calving ease EPD. The other point I wanted to make with this that I think hits home, that's a relatively large possible change, even on that bull that we would consider to be high accuracy for a yearling bull that has genomically enhanced EPDs, okay? So that's as high as we're going to get in this particular scenario, okay? So I guess as I evaluate that, to me, I think the thing to keep in mind is that genomics is a great tool. It's going to enhance our accuracy, but it's not going to replace progeny records in terms of establishing highly proven, predictable animals. We know that from the beef world. I think that applies to the sheep world as well. It will greatly help us on young animals, but it's not going to replace the highly proven stud that have several progeny on record over a couple years with much data, okay? We're not going to achieve that same level of accuracy using genomics. We have to have progeny for that to happen, okay? A few misconceptions as we kind of wind up here. Again, I'm going to use the beef analogies to share some things that I think we've learned. Number one, and this has been emphasized in the previous two seminar sessions, and I'm going to emphasize it again. We need phenotypes, okay? DNA collection and genomics does not reduce our need for collecting phenotypes. In fact, we could argue that we have an even larger need for phenotypes now, okay? Phenotypes on more animals to keep our training population robust and to better predict those traits which are sex limited, which are very important in sheep and occur later in life, okay? So those lifetime records are key and very, very important. The second one is that if we genotype, that's going to improve his EBV, okay? Well, in these talks, we tend to think about the EBVs that go up or are more favorable. Unfortunately, it works both ways, okay? The chance of them improving through genomics is equal to the chance of them actually getting worse, okay? So they don't all improve. Genotyping females is less important in males. That's a misconception, and we talked about that already, and I'll make the case again that as we move forward, we need to genotype females as well as males for those reasons that we talked about, okay? Only genotype the best animals, okay? That's a little like only turning in data, phenotypic data on your best animals, okay? That really doesn't help us, okay? To take advantage of this technology, we need genotypes on as many animals as we can, not just those select few, but a representative sample that represents all pedigrees and represents varying levels of phenotypes, okay? And lastly, the need for genotyping will decline after the reference population is established, okay? We need to continue to genotype. The reason for that is that our population of any animals is continuing to evolve as selection occurs. So population continues to change, okay? We want to have a reference population that is closely associated genetically with the animals that we're using genomics on, okay? So if our reference population is further away genetically from the animals that we're actually genotyping, it becomes less valuable to us, Okay, so that's the need to continue this as we move forward. So simple examples, how can we use it? We can differentiate twin brothers. Genomics will help us differentiate twin brothers. A little like that beef example I shared with you. Pedigree-wise, they're the same. They're going to start at the same point in terms of their EBV calculation, but genomics can help us separate them. We know they're full brothers, but we also know that genetically they are certainly not identical. Okay. It can help us with keep cold decisions on ram lambs earlier in life. Okay. Think about being able to make decisions on ram lambs as it relates to their ability to sire females or maternal traits that we're not going to realize for several months to years down the road. Okay. That's a huge advantage in terms of genomics. I would caution us collectively as a group that we continue to need phenotypes on those ram lambs. And so while we're going to have an increased ability to make some of these decisions earlier in life, and we say at weaning as opposed to maybe post-weaning, we still have the need for post-weaning records. And so we still need to continue to collect those. And then it can help us, as we talked earlier, wish issues or challenges on particular animals relative to their individual performance record or their contemporary group structure. Okay. So Genomics enhances our selection largely through increased accuracy. And that happens in several ways that we visited about. Phenotypes remain the foundation of what we're doing from a genetic selection standpoint. We must continue to collect phenotypes. The only way that DNA and genomics has value is if we have phenotypes to associate it with. Okay, we want to do that on both males and females. We've learned that from other species. And DNA certainly will not eliminate or replace our need for a good record book using our scales and all the other measurement tools that we have out there that we've traditionally used. It will just complement it. It will enhance its value and hopefully down the road pay us dividends in terms of profitability. With that, Andrew, I'd be happy to entertain any questions or if there's discussion.
Thank you, Dr. Greiner. Really enjoyed that presentation. There's a couple questions that came in through the chat over the course of the presentation. One, can you use this information to hone in on genes that are responsible? They specifically listed like high birth weights. Can we identify those specific genes with genomic technology, I think is what they were hinting at.
So for most of the economically important traits that we have, what we do know is that most of those are controlled by multiple genes. And so we have multiple SNPs that impact those particular traits. So unlike traits, for example, scrapie resistance or the myostatin gene, most of the causative genes associated with our economically important traits have a relatively small impact individually, but it's the sum of all those individuals that's important.
So then there was another question, and it relates to grading up or upbreeding individual breeds, like using an AI sire from overseas to grade up use here. And could you use genomic technologies to calculate the true percentage of that breed that I assume that you're trying to grade up? I think that's what the question was aimed at.
So I'll answer that this way, Andrew, and that's a little out of my wheelhouse to be honest, but I think through genomic technology we can establish differences in breed composition, and exactly how we use that in terms of incorporation into the EBV calculation, I'd have to defer to those with the expertise in that particular area. I don't know if that's a question to be emailed to Dr. Lewis or others, or if they're online and would like to interject, I'd happily entertain that.
If anyone else has any questions, feel free to put them in the chat, or you can unmute yourself and ask them.
So Andrew, I see quite a bit of discussion on here, of course, and as was pointed out in the previous webinars, the Katahdin breed, of course, has got a project underway, and currently is the only breed that has a reference population established, and they are in the process of implementing this technology. But those that's been discussed, that process, the protocols, the concepts that we've all discussed is applicable to all those breeds, and we can apply it to different traits. Wool traits was mentioned. It's the same process. We have to establish the reference population. We have to establish the SNPs of interest that relate to wool traits that are important. But that process is exactly the same. And fundamentally, those things that we've talked about apply. It's just the different breeds are at different stages in terms of where they're at with the technology and how we can currently apply it today.
Scott, if you'd like, I can try to maybe add a little bit on that question about looking at percentage of breeds.
Please do, Ron.
Yeah. Had to close the door there behind me for a moment. The challenge with using genomics in that way is that sheep have genes in common because they're sheep. And so there's a degree of similarity just because they're sheep, putting it quite simply. And so before we can actually try to disentangle whether a sheep is a percentage of one breed versus another, what we really need to do is we have to establish the genetic makeup of those individual breeds. And we have done that in a few cases to show that a Katahdin is distinct from a Rambouillet, that is distinct from a Suffolk. So the starting point is certainly to establish those differences, what make breeds different rather than similar. And if we can do that well enough, and that's an important part of this, then we can start trying to take into account in crossbreds what proportion of that crossbred relates to one breed or to another. In cattle, that's certainly being done in some of the crossbred composite breeds that are in play. In sheep, at least in the US, we probably don't have enough of foundation of really linking the genetic makeup to separate breeds to be able to deal with crossing very well at the moment. But hopefully that's someplace we'll get to in the future as we start developing more of these reference populations and we learn more about what make these breeds different genomically rather than their overlap. I hope that helps a little bit.
That was good, thank you. There's one more question in the chat that came up. Do the reference data for the breeds currently being compiled consist solely of NSIP enrolled sheep, or are there outside genetics to make a more random sample?
Andrew, I believe that it's currently NSIP enrolled because those are the animals that have phenotypes.
Yes, and that's my understanding as well. And I believe, there's, like Dr. Greiner said, you don't just genotype the best animals. Like you don't want to submit phenotypic records on just your best animals either. And so during this process, there was individuals that were genotyped and sampled, across the spectrum of the breed within NSIP identified through NSIP.
Andrew, I'd follow up on that and just say that within the Katahdin breed, in establishing that reference population, there were flocks from across the U.S. In many different environments, many different management systems, as well as within flocks that were sampled. In most cases that I'm aware of, the entire lamb crop was sampled. In many cases, the entire lamb crop was sampled. Certainly for mine, it was. So that was the duds and the studs all together. So we got a very, from my personal experience, we got a complete range of the performance and diversity in our flock here on the extreme northeast corner of the U.S. And there are other flocks, all across the U.S. So that's the level of genetic diversity that went into the 5,000 animal reference population for Katahdins.
Thank you, Tom.
Maybe another angle on that question is that without the 5,000 phenotypes that went into NSIP for most of the traits, there's no value to the reference population. The reference population has to have both, and it needs to be in an NSIP setting or similar. And it doesn't have to be NSIP, but there's no other group doing that in the US.
Any other questions?
This is Joe Emenheiser, just kind of following up on that thought and thinking about your comparison with the beef industry and the role of breed associations being different. I'm kind of looking for the pot of gold at the end of your rainbow there, and thinking about the genetic repository or a collection of data and the ownership of that. In the beef world, the breed associations take an active role in the collection of those data and the ownership of the data set and the evaluation of it. And we're different here. Thoughts on vision for how things will or should shape up in the US across these different breeds that are getting involved in the technology?
Well, Joe, as I mentioned, I think that's certainly a challenge. And I think as we think about that in terms of expanding the reach of NSIP, expanding the reach of utilization of genetic improvement tools, and specifically performance recording and use of EVVs, and associating that directly with a documented pedigree that's available and can be used. I think from a structure standpoint, it makes a lot of sense that breed associations become engaged in that. And I applaud those that have and are investing time and resources into advancing this technology and others. Not only genomics, but the whole principle behind genetic evaluation and use of performance records and use of genetic evaluations. And so historically, to be candid, we all recognize there's some hurdles there. And the sooner we can come together to work out how we get over those hurdles together, I think the quicker we'll move forward as an industry. I don't have a silver bullet though, Joe. I wish I did.
There was one more question that popped in the chat. How do we determine how many animals are required for a reference population? Is based on breed size. How do we go about determining that number?
So if my recollection's correct, Dr. Lewis had a slide last week, and certainly the larger and more robust your reference population is in terms of diversity of genetics and number of animals and number of phenotypes to associate with that, the better. My recollection from the data that he presented is that the 5,000 as it relates to Katahdin was at an optimum relative to that breed. So Oogie, I would reference you to Dr. Lewis's webinar last week, which is posted on the NSIP website. He had a couple slides which addressed that specifically and showed that.
Scott, if you wish, I can make just a couple comments
Sure.
on that, but I don't want to interrupt.
Sure.
What we're trying to do in a lot of our breeds at the moment as we do the evaluations is based on pedigree, because that's typically all we have. We try to look at pedigree relationship among our animals and estimate something called the effective population size. And that can be thought of as the number of independently breeding animals in a particular breed type. If we think about Holstein cattle, and Scott, you probably have a better sense of this than I do. I think the effective population size in North American Holstein is 40 or 50 independent animals, pretty tiny. From what we have done so far, and it is not complete, it's not complete, you know, we're finding effective population sizes based on pedigree in many of our breeds, somewhere between 300 to 500 animals. And what we've looked at so far would be really the Katahdin and the Suffolk as our main examples. My guess, and it's just that when we move to the Rambouillet, some of our Western range breeds, I think the effective population size is going to be larger. They're going to be genetically more diverse, which means you need a larger reference population to be able to do a good job with genomics. In breeds that have very few animals, that, you know, almost inherently, they're going to be relatively more closely bred, and maybe a smaller reference population would suffice. But what we're trying to do is to actually look at that to try to get a better figure of what would be needed for particular breeds. And then once we collect the genomic information, we're going back and verifying, if I may, are we big enough? Because we can then do it with the genomics. And that's something underway in the Katahdin.
Thank you, Ron. Are there any other questions here before we wrap up? So out of all the Katahdin breeders that are actively reporting, whose flocks were used to make up the reference population? Or I guess how many flocks were included in that reference population of 5,000 animals?
I'll comment on that, Andrew. If in the Katahdin Herald Fall 2020 issue on page 8, there's a list of all the flocks. That's the fall 2020 issue of the Katahdin Herald, page 8. There's roughly 20 flocks.
Thanks, Tom.
Yep.
And they were ones doing fecal egg count EBVs.
Good point, Jim.
Well, if there's no further questions, thank you for attending this evening. Hopefully you can all join us next Tuesday night for our final webinar in this series. We'll be covering the the sample collection and data submission process related to genomic-enhanced EBVs. So the registration information can be found on the NSIP website if you have not registered already. So hopefully you can join us next week for that. Enjoy the rest of your evening, and hopefully we'll see you next week. Thanks.